Hello everyone, welcome back to this third and final part of episode 8. In this episode, we will explore the activity-dependent wiring of the entire brain. Then we will take a look at some of the characteristics of development in childhood, adolescence, adulthood, and aging. Activity-dependent wiring means that the formation and pruning of synaptic connections are dependent on the activities. For example, at the cellular level, this means that there are Hebbian modifications. This is a classic quote in neuroscience: neurons that fire together, wire together. There are also establishment of correspondence patterns, how one neuron corresponds to another, or a few neurons corresponds to another few neurons. And postsynaptic feedback. The action of the postsynaptic neuron feeds back to the presynaptic neuron and influences its behavior. There are many other ways of doing this, of forming and pruning synaptic connections. On a more macroscopic level, cortical areas establish connections via activity, and moreover, connections are fine-tuned with activity. Let's first take a look at the cellular level. At the cellular level, a frequent occurrence is synaptic rearrangement, the formation and pruning of synaptic connections. For example, presynaptic neurons A and B now both have 3 synapses on the postsynaptic target cell. Now, after rearrangement, A becomes weaker. It now only has 1 synapse, while B becomes stronger, having 5 synapses. Let's take a look at an example of Hebbian modification. Firing together means wiring together. For example, if the neurons in the blue group fire together, then this neuron will also fire, because it has more synaptic connections with the blue neurons. If the red neurons fire here, then this neuron will also fire, because it has more synaptic connections with the red neurons. Then, after a while, the connection will become like this. There is synaptic rearrangement. Blue neurons only connect to the right neuron, and red neurons only connect to the left neuron. And now the cortical areas are separated. This is how firing together can cause wiring together. Another example of the synaptic rearrangement is at the neuromuscular junction. One characteristic of the NMJ is that a pattern is established. This pattern is that one neuron controls one or many muscle cells, but one muscle cell only corresponds to one neuron. Here, at first, there are several alpha motor neurons, and each neuron innervates many muscles. After maturation, one muscle fiber is innervated by only one alpha motor neuron. Moreover, the synaptic connections between the neuron and the muscle cell become more. There forms a lot of neuromuscular junctions, so that the neuron is capable of stimulating the muscle fiber by itself. There is no need for multiple synaptic connections with other neurons. Another characteristic of the NMJ is postsynaptic feedback. The reception of acetylcholine by the nicotinic receptors is critical for synaptic maintenance. If we apply a drug to block the nicotinic acetylcholine receptors in a small area, then there is loss of the receptors at the site of blockage. As a result, the axon branch at the site of blockage will be withdrawn. This is how feedback influences the neuromuscular junction. Another very good example on the macroscopic level is the primary visual cortex. The primary visual cortex has a specific characteristic called ocular dominance columns, like this. The black patches are dominated by one eye, and the white patches by the other eye. There is an alternating pattern between the left eye and the right eye. This pattern is formed via spontaneous activation of the 2 eyes during development. When our eyes have not yet opened, our left eye and right eye will already start releasing signals spontaneously. These signals are alternating. The left eye first lights up, and then the right eye lights up, alternating with each other. This facilitates Hebbian modification, and thus the visual cortex is divided into ocular dominance columns. This is further achieved after we open the eye. when we receive visual stimulus with both eyes, then the ocular dominance columns will form even more better. If we receive visual stimuli with both of our eyes, then the ocular dominance columns will become even more distinct and established. However, this is dependent on external visual stimuli. If we deprive the vision of one eye for a prolonged period of time at an early age, then there will be a failure of establishment of ocular dominance columns, as shown in the bottom figures here. There is not much distinction between the two eyes. If we cover one eye of a kitten or sew the eye together, then the kitten will fail to establish the ocular dominance columns in certain cases. Here is an example. We deprive the vision of the one eye of the kitten at an early age. Then the closed eye will become unable to provoke a response in the cortical neuron. It used to be like this before deprivation. Both the left eye and right eye can provoke a signal, though the right eye is stronger. However, after deprivation, the right eye is no longer able to provoke responses in the cortical neuron. Moreover, the open eye becomes dominant in terms of ocular dominance groups. Some neurons are completely dominated by the left eye, some others by the right eye, while some neurons are in between. Maybe the left eye has a stronger influence, but the right eye also has an influence, though weaker. However, if we deprive the vision in the eye, then the ocular dominance groups will become increasingly leaning towards the open eye. The closed eye will rapidly become unfunctional. We can conclude that deprivation leads to dominance of the open eye. Another condition, which also occurs in human beings, is called strabismus. It is, in vulgar terms, sometimes called cock-eyed or wall-eyed. In other words, the eyes are unable to align themselves. There is unsuccessful binocular vision. In strabismus, there are exclusive connections in the ocular dominance columns. Usually, the ocular dominance group is like this. There are few neurons that are dominated completely by one neuron, but more that are in between this spectrum. However, in strabismus, most of the neurons are dominated solely by one eye. As a result, strabismus will cause the loss of stereoscopic vision. In other words, with our vision, we cannot sense the depth, the distance between the object and us. We cannot see the three-dimensional aspect of things. Strabismus is a condition that must be treated at an early age. If otherwise, then this ocular dominance group and loss of stereoscopic vision will persist throughout the life. However, if this condition is treated at an early age, then we will talk about the concept called critical period later, and that will help us explain why treating strabismus can help correct vision. Modulatory influences have a large role in activity-dependent wiring. We mentioned diffuse modulatory systems in the episode about neurotransmitters. For example, the norepinephrine system originating from the locus coeruleus plays a large role in the activity-dependent wiring in monkeys' vision formation. If we have this modulatory influence and we sew one eye, we deprive the vision of one eye, then the ocular dominance group will experience a large change. However, if there is not the modulatory influence, then deprivation will not lead to a very significant change. Therefore, we can say that the modulatory influences are important for cortical plasticity. The plasticity of binocular connection is limited. Why? It has a critical period. Let's take a look at this experiment. When a kitten is at the age of 4 weeks, we deprive its monocular vision for 2 days. Then the ocular dominance column will become like this. If we do this experiment when the kitten has 10 weeks of age, then the ocular dominance columns will become like this. If we do it at 12 weeks, then something like this. If we do it at 16 weeks, then the ocular dominance groups hardly change at all. Therefore, we can conclude that as the age advances, as we become older, the plasticity becomes lower. Binocular vision is formed at a certain stage in our life, and later we cannot change it anymore. Therefore, if we correct something like strabismus before this critical period end, then the plasticity can help us correct the ocular dominance groups, so that the strabismus patient can become normal again if he is treated early enough. The critical period has a very good example. Imprinting is a behavior that epitomizes critical periods. Imprinting means that when a small young bird is born, it will identify any object or organism around it as its mother. It will follow that organism anywhere. Konrad Lorenz is an animal ethologist who discovered this imprinting behavior. He experimented with geese. When geese were hatched from an egg, he stayed around the little gooselings. When the gooselings saw Konrad, they identified Konrad as their mother, and they followed Konrad around for a very, very long period of time. Critical periods is not accessible for all mental functions or faculties. Not all of them have critical periods, but certain important ones do. For example, language acquisition is usually considered to have a critical period of 5 years. If a child does not contact any language in the first 5 years of his or her life, then he or she is unlikely to acquire language at a later stage in life. Then he or she is highly unlikely to acquire proficient language abilities in a later stage of his or her life. Similarly, emotional control usually have a critical period of around 2 years, and motor control has a critical period of around 8 years. Let's take a look at some of the other characteristics of childhood development. Growth. There is extensive growth of the nervous system continued into childhood and adolescence. There is fast growth of neurons by neurogenesis, especially in the cerebellum, which facilitates our learning of new motor skills. That's why children can learn motor skills much faster than adults. There is also synaptic growth, which helps us form new connections and form new memories. On the other hand, besides neurons, glia also grow a lot, especially the oligodendrocytes and Schwann cells. They provide myelination for the new axons. Myelination facilitates saltatory conduction and increases the conduction speed. This helps us improve our mental functions. It helps us gain higher cognitive functions. On the other hand, there is also retrogrowth. This is somehow surprising. In fact, retrogrowth may even be more important than growth itself. In retrogrowth, I mean that some things are pared back. For example, neuronal networks become smaller via apoptosis. Apoptosis is a process called programmed cell death. Some neuron cells initiate their own death program. They commit suicide. This is due to sometimes trophic factors. Trophic factors are factors that are essential for the survival of neurons. For example, the nerve growth factor is essential. A postsynaptic neuron provides the presynaptic neuron with nerve growth factor. If the presynaptic neuron does not receive the nerve growth factor, then it will initiate apoptosis and it will die out. This helps us eliminate unuseful neurons and pare back the neuronal network, so that our networks are much more efficient. There is also synaptic pruning. Just like pruning off branches from a tree, we prune off synapses from the axon. In fact, a 2-year-old has 50% more synapses than an adult, even though the cerebrum is only 80% the size of an adult's brain. Into adolescence. There is more synaptic pruning. There is also more myelination. That means white matter grows a lot, especially in the corpus callosum. That helps the left and right hemispheres of the brain to connect more frequently and more conveniently. There is also neuronal growth and better connections, especially in the prefrontal cortex and reward systems. That's how adolescents are beginning to get better at higher functions such as decision making, because these are the functions of the prefrontal cortex. The plasticity of the neurons remain high. There is also 2 types of plasticity: experience expectant and experience dependent. Expectant plasticity is expected of experiences. That means our neural circuits are designed to expect experience and to change with experience. It's not essential for our survival. However, experience will still change this experience-dependent plasticity. Into adulthood. In adulthood, neurogenesis is now limited. It only occurs at certain sites. For example, the dentate gyrus in the hippocampus. New neurons in the hippocampus help us form new memories. The olfactory bulb is a bulb where it receives olfactory or smelling information. Since these neurons are extremely susceptible to external influence and sometimes die out, we need neurogenesis to fill in the blanks. In adulthood, regeneration is also limited. Regeneration of all neurons is sometimes possible in lower vertebrates. However, we are a higher type of vertebrates, and so our ability to regenerate neurons is limited. In humans, in the PNS, we can regenerate some axons and neurons to a certain extent. However, in the CNS, we cannot do so. Why is that? One possible reason is due to the oligodendrocytes. As we know, oligodendrocytes are the glia cells that help myelinate the axons in the central nervous system. It releases a factor of no-go to prevent the regeneration of axons. This is kind of funny name because no-go is just a name that stems from no and go. It tells the axon you cannot go there. For example, if we take this experiment, suppose that our optic nerve from our eye is broken at a certain point. Then, since there is no-go in all of the nerve, we cannot regenerate the axon from the eye to the visual cortex. Then, since there is no-go all over the place, we cannot regenerate the axon from the eye to the thalamus. However, if we graft a peripheral nerve here, if we take a piece of peripheral nerve and put it there, then there is no-go in this nerve. The axon can grow almost to the thalamus. However, since there is still no-go inside the thalamus, the axon can never really reach the thalamus. This is an illustration of regeneration in PNS and inability of regeneration in the CNS. In adulthood, gray matter volume peaks in childhood, while white matter volume peaks in adulthood. In fact, the prefrontal cortex only fully develops around 25 years old. That is quite late, and that also explains why adolescents, although they are becoming better at decision making and at controlling themselves, cannot fully master this skill until they become young adults. Neuroplasticity is slightly lower in adulthood. However, we can still form new memories quite efficiently. Mike Merzenich, a scientist, suggests that exercise can actually improve neuroplasticity, even into the aging phase. So exercise seems very useful. Aging is different from dementia. Dementia is a kind of pathological process where there is significant decline of mental capabilities, as well as significant shrinking of brain tissue. Aging is different. In aging, mental decline is normal, though it is not to a large extent. There is decline of declarative memory, decline of working memory, but non-declarative memory largely remains. There is also a decline in certain types of attention, such as resisting distraction or doing two things at the same time. Also, physiologically, there is shrinking PFC, cerebellum, and hippocampus. There is also thinner cerebral cortex, as well as decreased thin spines and shrinking dendrites. Thin spines are a kind of dendritic spine that are extremely plastic. These spines help with neuroplasticity a lot. The decreased number of thin spines means that the neuroplasticity has decreased. There is also decrease in neurogenesis in the few areas that still have neurogenesis. Some possible reasons of aging include these. The regulation of gene expression is disrupted, or gene expression is changed. There is extensive oxidative stress. For example, free radicals occur in the brain. Free radicals are chemical substances that have unpaired electrons. They are very reactive and can disrupt the function of cells by disrupting proteins or other molecules. We usually have a mechanism of recycling and dealing with free radicals. However, this mechanism is not perfect, and free radicals can build up in our brain as we age. They can cause DNA damage and damage our cells. Another one is immune dysfunction. The microglia dysfunction decreases. There might also be inflammatory response inside the brain, which is bad for our brain tissue. Another possible reason is the buildup of abnormal protein and formation of aggregates. Just like free radicals, we usually have chaperones and other proteins that help deal with aggregates. However, they're not perfect either, and there is a buildup of abnormal aggregates. The buildup of aggregates is especially severe in neurodegenerative diseases such as Parkinson's or Alzheimer's disease. However, aggregates are also normal in normal aging brains. In this episode, we have talked about activity-dependent wiring, childhood and adolescence, adulthood, as well as aging. By now, we have covered all the major aspects of the development of the nervous system, as well as its development and changes into our old age. In the next episodes, we are about to start a whole new section, that is, sensory and perception. We will start with the visual system, which is the best example of those systems. Hoping to see you again in the next episode.